Let's move on to our second objective of this particular video, which is understanding sort of the purpose of both what goes on during the peer review process and then as a reader of the published work, how you would actually analyze this. And so overall, it's to improve the science. I'll say that again. We want to make sure we are better scientists, that the science is correct, that the findings are conclusive for us to accept the understanding that is trying to be conveyed before we all move forward and try to build on that work. Everything that's published is a puzzle piece towards understanding a big mechanism, whether that's how cancer works or how infection works or anything else that science is achieving. Every author's contribution is one piece of a big puzzle, and we all have to work together as a scientific community and as a readership to vet and do this together. So I always think the purpose is to improve the science, and we want to do that by identifying any potential inconsistencies with the method that you've been taught. So let's break this down into sort of the ways you can do an analysis based on the anatomy of a paper. So the first one we're going to tackle here is related to the materials and methods section. So really kind of vetting the methodology that the researchers used and the design of the study at large and each individual experiment as well. So right away, we're going to start with the materials. This particular section should give you a thorough description of all the solutions and reagents and chemicals that were used to perform the study. And you need to think about them very closely to see if they're aligned with what consistently is being used in your field. One of my favorite examples was an animal study that looked at the role of sugar in the diet in terms of how it modulated bacteria in the intestine and overall animal behavior and physiology. And I looked into the materials and I learned that to get the sugar into the animals, they actually used grape Kool-Aid. And I thought that was a particularly odd choice, but it was the choice they used, and it was not inconsistent with what other groups were doing. So you want to make sure that the materials are consistent, that you understand what they are, and that in some cases journals have provided the manufacturer that they purchase those materials for. And that becomes very, very important for you someday as a user when you might want to repeat a study or build off of a study and know, oh, where did they actually buy those materials? I know that as a reader, we're teaching you to read top to bottom this paper, and so that you understand how to do it. But in practice in your career, you might not actually be doing that in full. You might just be looking for a particular method or a particular material so that you can do your experiment. Or just looking at a particular set of results so that you understand one of them. Sometimes you don't have to vet the whole paper. Once you get deep into a particular field, you really don't even need to read the introductions anymore because you're so familiar with them, given that they're very consistent for a particular field. So first, we want to look at the materials and vet them thoroughly. Next, and very importantly, the choice of study model. There could be limitations and or inconsistencies with how the researchers set this up and are making conclusions. They're based on those particular study models. So for example, if a researcher is doing cell culture and using human cells that are in isolation of everything else and put into a dish and grown in media in a lab, and then making a big sweeping conclusion about human behavior, you should slow down and say, well, you are getting there. The human cells are one step of the puzzle, but you grew them in a dish, and so there's limitations between making that sort of statement and a much broader one, and I'm going to return to this in a little bit. Study models might be animal, and maybe your field of study uses mice. And you want to make sure it's consistent with other publications. If they start to use a different model, like a chinchilla, that might not make sense to you, unless, of course, you're studying middle ear infections, in which case chinchilla, believe it or not, is the study model of choice. And if you don't know what a chinchilla model looks like, or what a chinchilla is at all, I encourage you to do a little Google image. They're really cute, little furry creatures. So you want to vet the study model, whether that's a benchtop in vitro experiment for community behavior of bacteria cells or anything else you're doing. You then want to look and make sure the sample size is not only consistent with your field, but also consistent within a paper, and especially consistent within a particular figure. There are obvious reasons sometimes that a sample size might vary. So for example, you started off with 6 mice and in group 1, and then you had 6 mice in group 2. Let's return to the week 3 biomath example where you had treated and non-treated animals. And maybe the treated group, 1 died off, and so you went from 6 to 5, but it's still a significant amount of animals to run with the final result. You might see it vary then from comparing 6 to 5. Not apples to apples, but we can roll with it. Sometimes it's very inconsistent and you can't. So you want to make sure that there are a correct number of biological and technical replicates, and we're going to teach you about what those mean both this week and in subsequent weeks, and that it's consistent with your field. So you have to evaluate the sample size. You also want to make sure that the study was designed with all the appropriate controls, and that they are represented in the data set at a satisfactory manner so that you can actually believe in the results. Controls, both positive and negative controls, are extremely very important. A well-controlled experiment gives you the trust that you need to think that the result is one that you can build off of or not. For example, let's go back to our treatment versus non-treatment of the mice. Let's say we were treating with compound A, and that's our treatment group. Well, compound A may have been dissolved in a buffer, let's say ethanol. So compound A was dissolved in ethanol and given to the treatment mice. The vehicle control for this experiment, the negative control, would be ethanol by itself. We want to make sure that the compound that we're testing, compound A, is giving the effect that we're looking for on the animal one way or the other, and not the thing we diluted it in. So ethanol in that case would be a control. The second component of the paper, the anatomy you've learned that we really need to vet thoroughly, is the results. And there's a lot of ways to do this, just like there are the methods, and we're only giving you a couple. And one of the key ones that I like to start with, especially at this stage when you're learning how to do this, is looking for agreement or lack of agreement between what the sentence says in the results section and then what is actually represented and shown in a particular data set, whether that be a figure or a table. Believe it or not, there are inconsistencies, and we're going to task you with finding some on your own this week as you're analyzing the paper of choice. So in other words, let's say a sentence says that a result is significantly different between 2 groups. And that's what the result says in the sentence, okay? And then it references, let's say, figure 1. So you turn your attention to the data, and you want to look for an asterisk with a p-value indicating statistical significance. If it says that the 2 groups are not different or they're the same, then you should see a lack of statistical significance. Sometimes the sentences don't match, or sometimes, believe it or not, the sentence will say the result shows an increase, and then you look at the data and it was actually a decrease. This happens too. So you want to look for agreement. And then, as a bonus, you kind of want to look to see if there's anything else in the data that might not look consistent. For example, they might want you to look at the vehicle control versus the treatment, and let's say it goes up significantly, and they say, oh, great, it's significantly increased. And then they do a knockdown treatment, and it goes down significantly from the increase, but it's still significantly different than the vehicle control. Those differences between the knockdown and the vehicle control could also be important. And that's not necessarily a deal breaker for you to believe in the work, but that could also lead you to new questions that generate new experiments for future directions. We want to know now, why is the knockdown not quite the same as the vehicle control? Maybe there's an important difference there too to understand. And I think that evaluating the results to do this in part, which is to identify future areas of study, is a really important tool for you to learn. And I think it actually makes science more accessible. I always thought that to be a successful researcher and scientist, I would have to develop Nobel Prize-worthy ideas and questions and hypotheses. And while there's absolutely a certain element of designing your own, I then learned and took a lot of comfort in the fact that the literature, the papers, the results provide a needed framework from which to springboard from. And so I wasn't locked in some room all by myself trying to think of all this by myself. I was actually using the papers to generate a springboard, a platform from which to draw new experiments, new aims, and new directions, and build off of things that I thought, wow, that might not be a critique necessarily or a fault with the study that I'm analyzing this paper, but this could be a direction that they might not be thinking about going in that we could take our work. And so it actually made it feel a lot more accessible to me. You always want to look at the axes / the scale on a graph, or any other way that the particular results are presented to you, any other parameter in a data table or otherwise. And so the first thing I always do is ask myself, what's on the y-axis and what's on the x-axis? What are those scales showing me? Is it percent increase? Is it value A versus value B? Is the x-axis at a time scale? Am I looking at, let's say, growth of bacteria treated and untreated by a compound in a test tube over a course of 12 hours? What am I actually looking at? Let me understand the results, and let me make sure that these axes, scales, or other parameters are consistent with how other biological studies in my particular field are done. So for example, if you do a growth curve of bacteria, and let's say the majority of the field does this over a course of 24 hours, but this group does it over a course of only 10, you might be left to conclude that the authors should continue the study, redo the experiment, and complete the full 24 hours to be in alignment with everybody else. This takes a lot of time to realize the other works in the field, so don't be afraid that you didn't know that or anything else related to any particular field. Once you're in there, you'll start to feel that comfort level increase. You want to make sure that the data are visualized appropriately, and we're going to have a whole week on this to come, and you're going to learn how to do this, and you've kind of thought about it a little bit already. But you want to make sure that, let's say, bars or individual data sets, numbers of things are consistent and presented in an appropriate manner that aligns with everything else that you know to be true within a field. And of course, that the choice of statistics is appropriate, and the way that the data is analyzed to get those statistics was correct, whether that was an ANOVA or a t-test or any of the other ones you've learned. You could fold this into the methods as well. We could have vetted this there, and that would have been just fine too. Or you could vet it within each individual result. A good result will potentially tell you the way they did the stats in the figure legend, as well as the method section. So there should be some helpful redundancy, I like to say, in the choice of the stats and the way the data was visualized, both within the methods and within the figure legend of the results themselves. So you can always make sure that those are done correctly. In other words, did they correctly choose a one-tailed or a two-tailed t-test, or should this have been a completely different method of analysis altogether? While the majority of peer review for publication and for you as a reader to vet scientifically in your analysis of an article, while the majority is done in that first two components, the materials and methods sections and the results, we also want to consider whether or not the introduction and discussion sections also comply. And so this becomes a little bit more broad than the nuts and bolts of sort of experimental design and what a graph is showing you, and instead becomes an exercise in placement within the literature that's been established for a particular field. And so a good introduction and a good discussion have enough references to other works to do that contextualization. And so you want to make sure you're citing supporting evidence and sometimes conflicting evidences that place your work within the correct context. It's important for a lot of reasons. We want to make sure that we understand the novelty or lack thereof necessarily of a findings. We want to acknowledge the previous works that are done by other groups that have led to yours. As you learned when reading the paper, a very good introduction section sort of establishes the significance of the work and then references the previously done work that led to your question, and so you get a feel for all those things. And a very good discussion will then contextualize their results with a nod to the field in terms of where the work could go into the future. So you want to make sure you've done all those things. And I always give a little plug here for referencing methods in particular. I think we always think to reference sort of the idea or the result, but sometimes not as much the method. And sometimes people will just reference a paper in which the method was referenced from a different paper, and then you go down this reference rabbit hole trying to find the original place the method was referenced, or I should say actually established. And so try to go all the way back and find that first establishment of a method and reference that one instead of a paper that came afterwards referencing that. That will be very, very helpful to any reader. We also want to make sure that the wording is consistent. You oftentimes in science find things that are overstated. Remember that a study done in mice is indicating results for mice, not humans. So make sure your understanding and appreciation for where it is appropriate. This was a hard lesson for me in graduate school when I did this experiment looking at gallstones and salmonella in a test tube, and I thought I had cured all humanity of a particular disease, and my advisor said, "Yo, you just did something in a test tube." There's a lot of steps between this and human beings and clinical trials and everything else that would go with that. So just know where the work was done and whether a result is suggestive of something, and we're going to go into specific wording in the next video, but or whether it's actually conclusively indicating something. And importantly, focusing on correlation versus causation, making sure authors are using the correct wording to describe their results. We're going to go into this further in the next video as well. And remember, if all this is done, this should be happy, this should be joyous, and this is for the whole global community of scientists and non-scientists alike. I always believe firmly that the job of a biologist is to communicate the results with the scientific community, but also with the non-scientific community. An appreciation of your work at both of those levels is not easy to do, but is necessary to make the scientific enterprise continue, so that grants are funded and papers are published. And in your discussions in class, your instructors might potentially start to tell you a little bit about sort of the different payment models for these papers becoming published, what a journal subscription fee looks like versus open access or not, And those things become very, very important when you think about getting this out there to the planet. So this concludes the first of the videos this week, and please feel free to move on to the assignment that's associated with it. Thank you.